Okay, so this new chapter then, we're going to look at an extension of what we were talking about last time. Remember last chapter I mentioned that DNA doesn't do it just to do it. DNA has to be told when to do it, how to do it, that kind of idea. And so that's the idea of when is DNA going to show up in making the RNAs and therefore your polypeptide. So the author is going to spend quite a bit of time differentiating between prokaryotes and eukaryotes. That's okay. We're going to start with the prokaryotes, dealing with something called the operon, and then move into eukaryotes. But both have to, again, know when to do it. You don't want to waste resources. You want to control when you make something. And so with that, natural selection, of course, has factored into this. It's devised a system that works well for both prokaryotes and eukaryotes, bearing in mind the fact that the only thing you can bank on is that at some point the environment will change. And when that happens, hopefully somebody somewhere in the population can handle the change. This is why you want new and novel individuals in the population. But generally speaking, then, it is designed a system to say you're going to do it when you need to and no more. Your cells, therefore your body is really good and not wasting those resources. So we're going to focus first off on the operon system. That to me is a bacterial system that again tells it when to make something. So notice it was saying negative feedback. There it is, feedback inhibition. That's what this is. You start with something, you go through a chemical mechanism. All these genes have to work to make the proteins, in this case enzymes, to get the final product, tryptophan. Once you've made tryptophan, then tryptophan itself goes back and shuts the system down. So when you have a high enough concentration of tryptophan, you don't need to make any more tryptophan. Now that'll continue to happen until the concentration of tryptophan drops down low enough, at which point the system could start up, make more tryptophan. When you have enough tryptophan, again, it shuts the system down. So it is a lovely system in that regard. Now this is a negative feedback. Bear in mind, there are cases of positive feedback in the body. But in this case, we'll focus on negative feedbacks. So I mentioned the word operon. Operon is like the umbrella word. Within that, it's a controlling system. You have this switch, it is the operator. You have the promoter, it's like, start here kind of idea. And then you have the gene that is going to regulate. And so you have two kinds of operons we talk about, a repressible operon and an inducible operon. And of course, you have molecules that are going to come by and help the system out in order to make sure, again, your DNA turns on when it has to, turns off when it has to, that kind of idea. And not uncommonly, Escherichia coli is a model that's often used. In this case, we're going to show it with things like tryptophan and even the lac operon. So notice with tryptophan, tryptophan is by default turned on. So it is a repressible operon. It's on, it's on, it's on until it's turned off, which is why we use that phrase. Turned off, it can be repressed, a repressible operon. And like I was just saying, that happens when the levels of tryptophan are high. You have enough concentration, so you don't need any more. Shut the system down. So it looks a little bit like this. You have the tryptophan gene, it goes and makes this repressor, but notice the shape of the repressor. It can't fit into the operator. So because it can't fit here, the promoter can function. The polymerase can do what it does. It goes down from the promoter region to the genes and it codes out these genes and you get your transcript. Once you have your transcript, you can go and make your polypeptides. Great, and you'll continue to do that until you've made enough tryptophan, the co-repressor. Notice now when you have enough tryptophan, it binds to the repressor. Once it binds to it, like we've been seeing all semester, it changes the shape. Once you change the shape, now it can bind to the system. And once it's bound to the system, it can shut the system down. The promoter can no longer have the enzyme do its job. It can't go down, make the transcript. You can't make any more tryptophan. That will continue, of course, until your concentration of tryptophan is low enough that it can no longer shut the system down. So it's, again, a pretty direct system. So like I said, the tryptophan operon is a repressible operon, by default turned on, but it can be turned off. So I thought I would show that to you. Now let me change over to my sound. Okay, so again, I can't say anything during this, but you can hear the video.
The tryptophan operon, TRIP operon for short, is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The TRIP operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRIPR, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the TRIP operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRIP-L, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRIP synthesis pathway. TRIP-E and D for anthranilate synthase, TRIP-C for indole glycerol phosphate synthase, and TRIP-B and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRIP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the LAC repressor, the TRIP repressor does not stick like glue, but periodically dissociates, allowing a low basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter two tryptophan codons in a region called 1. They wait for TRIP-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the TRIP tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
The main reason why I love Grammarly is I'm a uni student.
And of course, right into a commercial. The bane of my life, commercials. But I hope you enjoyed the video. So that's the TRIP operon. Now, that's one that's on and can be repressed. Another one is a LAC operon. There it is, LAC. That stands for lactose operon. This is inducible operon. That means by default, it's turned off and then gets turned on. In this case, we're still talking about E. coli. E. coli doesn't really like lactose. It prefers other sugars. But, you know, it's better to eat something than nothing, right? So it'll eat lactose, and specifically its isomer, allolactose, when it has to. So notice in this case, lactose is absent. And because it's absent, you don't really need this. So the repressor can function, it binds, shuts the system down. But when lactose is present, which means its preferred food source, something like glucose, is not really there or in high enough concentration, then that isomer, the allolactose, binds to the repressor. And now notice it's the wrong shape. It can bind. So now it's turned on. It's been induced to function. It's turned on. And it'll go and make its transcript. And by the transcript, it'll make its actual polypeptides that it needs to, for the proteins that it needs to make. So it's inducible operon. It's one that can be turned on. So in that case, I also thought I would show you a video here. I found a fun one that I thought that you might like. And again, let me turn on the speakers for you. Okay, hope you enjoy it.
Faster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells. Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. We want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase. And poor RNA polymerase cannot move forward and no mRNA can be made, therefore no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are three genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around, bacteria want these enzymes to be made so that they can use them to break down the lactose sugar. Then they can metabolize it. Dead bacteria are happy bacteria. Here's the repressor. There's actually a gene here that codes for producing the repressor. See this gene that we call I? It has its own promoter. Well, this gene codes for the production of the repressor. So why do we need this repressor? Well, it's wasteful to make things that you don't need. If there's no lactose, it wouldn't make sense to start making enzymes that work together to break down lactose. It would be a waste. The enzymes would just sit there. So if lactose is not present, then the repressor binds to the operator, and this blocks RNA polymerase. So mRNA cannot be made, and therefore the proteins, the enzymes in this case, cannot be made. But if lactose is around in the environment, something pretty cool happens. The lactose, remember that's the sugar, binds to the repressor. This changes the repressor's conformation. Try as it might, the repressor cannot bind to the operator. RNA polymerase finds its promoter, binds, and transcribes to make the mRNA from the genes on the operon. That mRNA will be used to make enzymes to break down the lactose sugar. And bacteria like to eat, so that makes them pretty happy. We have to say that we think it's pretty impressive to think about all the gene regulation that occurs in cells. And if you find it fascinating, Know that there are careers that focus on gene regulation and understanding how genes can be turned on and turned off. Because by doing that, we can gain a better understanding of a variety of diseases that have gene influences in the human body. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so I hope you enjoyed that. Again, so there are two different kinds of operons, the repressible and the inducible operons. So repressible is more or less like the tryptophan one, and the inducible is the lac operon. There are slight variations to this. You can see it here. This is the CAP version, quite a word, catabolite activated protein system. This is like amplifying the lac operon when it's there. Here, cyclic AMP is going to be used. There it is, CAMP. You've seen that a few times in the course. And when it's available, it kind of makes it like more efficient for doing the lac operon. But the moment that glucose is around, again, glucose is a more preferred food source, then you probably don't need it. So you can kind of see that here. Notice lactose present, glucose scarce. CAMP is also present. So when CAMP binds, it makes this more efficient. Again, the allolactose is binding so it doesn't shut down the system. So if it doesn't shut down the system, you're able to process more of the lactose. Again, allolactose is an isomer. Here we have lactose, but now glucose is present, CAMP's low, and because CAMP doesn't bind, you're still going to go and make the transcript, which means you could still process lactose, but it's just not going to be doing it quite as efficiently or as high a level as it would have been. Okay, so again, that's prokaryotes and how they do what they do. It's pretty direct. Eukaryotes a little bit more complex than that system. So with eukaryotes, then we have this idea called differential gene expression. Remember, every cell in your body has the exact same instructional set. We have no difference in the DNA found, say, in a pancreatic cell versus a cardiac cell. It's just how you use the instructional booklet that's a little bit different. And so there must be something within those cells that regulates how you're using the exact same book of instructions. And you can see here, there's a lot of steps that does it. So the overall big picture then, a signal comes in. We've talked about cell-cell communication before. Chromatin, of course, it has to unwind. As it unwinds, it gets down to the decondensed version. You make your pre-mRNA, you process it, the 5' cap and the poly A tail goes out into the cytosol, finds a ribosome, translated into the polypeptide. Of course, remember, that's just the primary sequence. You have to make it into the secondary and tertiary structure, then it can go up and do something, whether it's in the cell or out of the cell, one of the two. So this idea of how tight this is wound, remember that affects DNA. The more tightly packed it is, up to and including a chromosome, it doesn't do anything for you. So there's two things here, acylation and then methylation. Acylation is loosening the DNA. And as you loosen it, there you go, loosen right there, that's right, you're able to do something with it. Methylation, methyl groups, there you go, that's that word I've been using, tightens it. And as it tightens it, you're less likely to use it. And that's known as this. Now, remember what I said about DNA. DNA is wrapped around histone proteins. There's 4 proteins on the inside and 1 on the outside. To get DNA to work, you have to get the DNA entirely off the histone proteins. And then you can transcribe it and later translate it. So that's what this is. You can see those 4 histone proteins that DNA is wrapped around. This is that nucleosome, the beads on a string we talked about before. It doesn't really show that one histone protein on the outside, but that's okay. All they're really getting at is if you have something tightly wound and you acylate it, you're going to decondense it. You still have to get it off the histone proteins, though, for you to be able to use the DNA. Methylation goes the exact opposite direction. So methylation, again, tightens it. And as you tighten it, you're not going to be able to use it. That's why they say long-term inactivation. Notice this goes all the way back to the beginning. You know, what's turned on, what's turned off, coming from mom and dad's alleles does get impacted here. And so impacts, you know, your embryogenesis and who and what you are. So we have then structures that affect the chromatin, affect whether it's going to be tight or going to be loosened up, and therefore whether or not you're going to use it, whether or not it'll be able to function as a coding structure. So you have these things called control elements. Remember, most of the DNA that's out there is non-coding DNA, and then you have to make this whole complex in order for DNA to work. So you have these control elements. These are the non-coding DNA that will affect what's going to be used for transcription, translation, that kind of idea. So you have to have like this whole complex created, and then you have to have the right elements evolved. Then you have to have loosened DNA, and then you're able to do something with it. So these are the essential components in order for you to transcribe something and then later translate something. So there's a lot of regulation that goes on here. So you have two things. Proximal means up close, distal means far away. Enhancers, these are like little molecules that will affect what's going to be coded, not coded. And then you have the activators. These are domain regions that serve to, again, to turn on, turn off the DNA. And so it's just a whole complex that you're making here in order to turn it on and turn it off. Like I said, not only turning on, but this is turning off, of course. You don't always want to use the system even when it's on. You don't want to waste your resources. So we're still affecting what's going to be tightening the DNA or loosening the DNA. So here's the enhancers. It's an area, again, upstream of the promoter. There's a promoter in your TATA box. Activators are molecules that will be able to bind to the enhancer. Once you bind to the enhancer, here's all those factors I was just talking about, creating this machinery. Once you have the machinery, then you have, of course, a polymerase bind, and then you can go and code out your transcript. So there's a lot of parts, moving parts here to get something to function. Now, really, again, these enhancers are really important. Notice it depends on what activators are available as to what's going to be used. You have pretty much the exact same enhancers pretty much in the system. So that's not a problem, but it just depends on the activators there. And so you can see different activators in the liver versus different activators that are found here in the lens of your eye. And you get two different things made. You have albumin, which yes, is called chicken white, that is correct, but it is a protein that's used in the liver versus, of course, this crystalline material that's used for the lens of the eye. Obviously, you wouldn't need this in the liver, and you probably don't need this in the eye. So it all depends on what's there as to what the DNA is going to do. Turn it off, turn it on, that kind of idea. So the eukaryotic version is a little bit more complex, perhaps, than the eukaryotic version. It's not just like the operon. Again, it's just control. There's a lot of genes scattered across the chromosomes is right, and it all depends on those activators that are there that'll help to use this part of the DNA. Okay, and once you actually make something, then of course we have our post-transcriptional modification. It means just that we've made our transcript, and you have to go and process it, put stuff on, take stuff off, that kind of idea. We've already mentioned this one, alternative RNA splicing. Remember, just because you have a segment of DNA, you can use the introns and exons a little bit variably. In one case, this might be an intron, and in another case, it might be an exon. I know I introduced this, but here you formally have it. So here's an example of that, exons 1, 2, 3, 4, 5. You made this, so you can see those exons 1, 2, 3, 4, 5. But notice it gets processed differently. So in this transcript, obviously the 4 didn't make it, so you're not using that. It's effectively an intron. Where in this one, the 3 is effectively an intron. So remember what I said about humans. We used to think, oh yeah, look at us, we're so amazing. We have 100,000 genes, and we don't. We've got about 20,000 genes, about the same as a mouse. And so it reminded us that we can do a lot with a little. How you process the DNA is a little bit variable. But at some point, again, you got to break it down. I've already talked about this as well in last chapter, the untranslated region. So you have the 5' cap, the transcript, the UTR, and then the poly A tail. That kind of sequencing, and then that this thing would sort of affect how long the mRNA stays around in the cytoplasm. The longer it stays around, the more polypeptide you're going to make. So again, you want to regulate when you turn it on, when you turn it off. Again, you don't just want to do it all the time. So a lot of this goes back right to the beginning. You have syngamy, we've talked about that before, fertilization of an egg, and then you have the division of the structures. So from one fertilized cell, it's a zygote if it's now developing, to 2, 4, 8, 16, 32, 64, 120, divide. And then as you're doing this, you're already beginning to form axes, and cells are beginning to realize where they are based on the axis. And as the cells talk to each other, they begin to realize what they're going to become. So we'll come back to this momentarily, but it goes all the way back to the very, very beginning. And of course, you have to break things down. So here you had a protein. Notice this thing called ubiquitin comes in. Proteasomes will then help you to destroy it. So you can see you break down the protein, and then you can maybe reuse the fragments. You don't have to necessarily ruin the amino acids, but just break down the protein and stop it. Now, there's a lot of regulation here. There's just a lot of acronyms. Sorry about that. So you just kind of have to learn the acronyms. I'm not going to hammer you too much on them. Just kind of be aware of what they are. But we're still talking about non-coding RNAs. Remember, a lot of DNA is not coding, so that makes sense. And a lot of RNAs may not be coding. The ncRNAs, there's a whole slew of them. You can see them here. mRNAs, microRNAs. Notice they block translation. Something very, very similar are RNAis. So interference RNAs They also get in the way of gene expression. Your author notes here again that they're very similar to each other. They just have a little bit different starting point. That's all. That's all you need to know about them. So we have the mRNAs, the RNAis, and then you have cRNAs, Si, not C like the letter C. These can block the chromosomes as well. And then you have P-associated pRNAs. Notice what they do. They induce heterochromatin, which means makes it more tight, and so it blocks it. Notice it blocks expression of parasitic DNA. Parasitic DNA jumping genes, transposons, can lead to big problems. So that's probably a good thing to block in that kind of regard. So you can then regulate how DNA does what it does by these RNAs. The RNAs can indeed be regulatory, when to turn it on, when to turn it off. This is huge, though. Notice as you copy the mRNAs, it allows for this morphological complexity. Comes up a little bit at the end of this chapter as well, certainly a lot in Bio 2. There's something known as the homeogenes, homeobox, or these homeotic genes. These are transcriptional factors that impact how many things you're going to have and where you're going to stick them. So it allows things to become more complex over time. Bacteria, generally not thought to be very complex organisms. I would remind you that biochemically they might be very complex, but in regards to, just a lot of stuff dangling off, no. But then when you get towards eukaryotes, yeah, throughout all of eukaryotes, whether it's protistans or plantae or fungi or animalia, it's getting more and more complex all the time. And then a little bit of evolution. They think the cRNAs came first, and then the Ms and the Ps. So again, just kind of a neat little sequencing of these regulatory RNA molecules. And you'll talk more about this, not so much in Bio 2, but probably when you get to your genetics class. Okay, so when I was talking about fertilized eggs, yes, so it goes all the way back to that. The fertilized egg, and then it's divide. So 2, 4, 8, 16, 32, 64, 128, it's divide. I didn't show it to you before, I don't think, but you end up what's called a morula. That's a solid ball of cells, which goes to a blastula, then a gastrula, and then a neurula. Just get you a nice little image here, something like this. Probably be fine, so I can make that a little bit bigger for you. Yeah, so there's your fertilized egg, and then 2, 4, 8, 16, 32, that kind of idea. And you end up with a solid ball of cells. The solid ball of cells then ends up being this fluid-filled cavity with a blastopore. It's a plug that's here. It's not shown in this image, but it would be there. And then notice that the cells are beginning to move and invaginate. This is called an archenteron. As you're forming this, you get to the gastrula, but the gastrula has embryonic layers forming, and you have 2 to 3 of them, whether you're called a diploblastic or a triploblastic organism. Truly the only diploblastic animal out there are cnidarians. Those would be the scyphozoans, hydrozoans, cubozoans. So that'd be like jellies and anemones and corals and the box jelly and hydrozoans like Physalia, which is man of war. And they only have ectoderm and endoderm. They don't have mesoderm. And interesting in this image, just randomly pulled it out. That's what you see here, only there's 2 layers. There can be 3 in a triploblastic organism. So let's show gastrula. Triploblastic. Triploblastic, there you go. Images. Okay, so again, that's not really showing it nicely. Sometimes it's hard to find a nice image. This is going to show it. It's just not the prettiest picture. But there you go. Pretty much every other animal, for example, on the planet has all 3 layers, triploblastic organisms. So you have ectoderm, outer layer, mesoderm, inner layer, middle layer, and then endoderm is the inner layer, and then you'd have like a gut tube, that kind of idea. And there's that blastopore that I was talking about. So that goes all the way back to this. When you're starting to develop the system, you're dividing, you're going from that morula to the blastula, the gastrula, and even that neurula that I noted to you. So the neurula would be after this. That would be even kind of nervous tissue that you develop out. I'm waiting on Teams. There it goes. So here, the cells are already beginning to position themselves and figure out where they are on the axis, therefore, where they are in relation to each other. So what are they going to become? How are they going to change from this undifferentiated cell to a differentiated cell? They're being induced to become something specialized. So like this, where are they in relation to each other? Here's that 32 cell stage. And so they're already talking to each other, chemically talking to each other, like we talked about back in chapter 11, cell-cell communication. And so cells will become fated. They're determined, and then finally they're fates. There's 2 main organism types we talk about, protostomates and deuterostomates. It's kind of old terms. Protostomates got changed into lipotrochozoans and ecdysoans, and then of course deuterostomates. He'll talk about these things in Bio 2. The old protostomates, again, which became the lipotrochozoans and the ecdysons, they fate early. So their cells are going to become that, whatever that is, early on. The deuterostomates, which are echinodermata all the way through the chordata, like you and I were chordates, we fate late. Our cells are going to become whatever that is, like a kidney cell later on. Because of that, actually, this is the reason we have identical twins. Very early on, if that embryo breaks and you're a deuterostomate, you still have everything you need because you didn't determine and fate early. But if you're protostomate and that happens and you fated early, that can mean the death of you. So this is a big deal. When's your cells fate to become whatever they're going to become, whatever tissue type they're going to become part of. So that's pattern formation. This again is, where's that access? Where are you? Are you going to be a toe, a finger, a nose, like for a human, that kind of idea. So your body's already dealing with this XYZ axis, and then where are you in relation to yourself, the axis, and your neighboring cells? So here, lastly, at the end of the chapter, here's the homeotic genes. These are the genes that deal with transcriptional factors. So really complexity, how many things you're going to have and where you're going to stick them, that kind of idea. And we've messed around with this, especially like early 20th century. They had some really intriguing experiments. I'll leave the how you feel about this kind of experiment to you. But this, again, is what you should have gotten. This is what they did get. This is like on Drosophila, where instead of getting the antennae, they were able to graft legs there. So once you're fated to become that, you're going to become that. And I've seen, not just with insects, I saw it with reptiles before, birds, kind of idea before as well, where they did some intriguing experiments. I'll leave the feelings behind that to you. And so last but not least, I told you pretty much at the end of all these chapters, they're going to get back to cancer. Again, it's just cells doing what they do. We still have difficulty understanding, why is it you have a cell that's a normal everyday cell, a cell that normally divides, that's just doing what it does, and then becomes a cancerous cell. So like an oncologist is a blood doctor, hematologist that studies in these cancer cells. So why is it? A proto-oncogene is one that's just a normal everyday dividing cell. Its DNA is functioning properly, and then something happens and now it's not. What happened? Why did it do that? Was it just blind random luck? Was it a mutation? What caused the mutations? Again, this is just something we're still working on. It's very difficult to know who's going to get cancer and who's not. Obviously, don't do things that might cause cancer. Don't smoke, don't drink. Try to avoid bad foods, get good sleep, keep your stress down, this kind of stuff. But again, a lot of things can do it. Rewriting DNA, a lot of things can cause cancers to develop. Again, you can have the best lifestyle ever and still maybe get cancer. So just all you can do is try your best, right? But at some point they might mutate, and now you have, of course, developmental cancer. It could be argued that every one of us has cancer in our body. It's a normal state. Cells just go wonky. You want to have a system in place to find those cells, hunt them down, and destroy them. Better to lose that cell than to have it proliferate. You don't want developmental cancer, obviously. So it's the same thing. Why are proto-oncogenes, the ones that aren't cancers yet, what becomes them? We have thoughts on that. I mean, here you go. Your DNA gets swapped one place to another, maybe like the parasitic genes idea, or extra copies are made in the DNA, or there's your mutations. Remember, don't define mutation as good or bad. Mutation is just a change to the DNA. The vast majority of mutations are silent, but still some mutations may be deleterious. So you've got to be careful with that. So like I said, you want a way to your body to have a way to keep this in check. So good sleep, keep your stress down, good diet, exercise, that kind of idea. Understand your diet changes throughout your whole life. So go those yearly physicals, that kind of idea, and give your body what it needs to fix problems when they happen, to make sure cells aren't misbehaving, and then when they do, deal with them. Better again to lose a cell than to have it proliferate and cause a problem. And of course, if anything is not right for you, have it looked into, that kind of idea. So cancer is known as the multiple hit model. Generally, no one thing causes cancer. So A happened, then B happened, then C happened, then D happened. I think the quip I gave, which is never meant to be a joke, was something to the effect of, one cigarette probably isn't going to cause cancer. That'd be like the worst luck ever. Could it? Yeah. So don't smoke, that kind of idea. So you don't want to put yourself in a bad position. There's never been a good argument for smoking ever. I heard one person argue about, well, people who smoke tend to lose weight. There are better weight loss strategies than smoking. So we know that if you smoke, you will most likely get cancer, that kind of idea. Again, could one cigarette do it? Theoretically, yes. So don't do it, that sort of idea. Alcohol, same thing. Is it healthy? Is it not healthy? There's a lot of research still going into this. So we're working on that. It's generally no one thing, but could there be one thing? Yeah, I mean, you get a mega dose of gamma radiation, you're not turning into Bruce Banner and the Hulk, you're going to die, probably. So again, be careful. And that's happened before. Chernobyl, there were people there that had apparently no problem and died of dispersed cancer within 2 weeks. 2 weeks after that Chernobyl event in 1986. So again, be careful. Be very careful out there. Just try your best, yeah? And then, of course, on the DNA level, it's hard. We got to make sure we have these tumor suppression genes working as well as we can. So, for example, these guys, the BRCA genes, just because you have the BRCA gene does not mean you're going to get cancer. It does not mean you're going to get breast cancer. Now, bear in mind, every human has breasts. It's just a trait coded into the vertebrate embryo before you either continue to be female or you offshoot to the male line. So we all have breasts. We all can get breast cancer. Now, my mom had and died of breast cancer. Does that mean I'm going to get it? No, but it means I'm at higher risk of it. Actually, one of my doctors actually recommended recently that I get specifically tested to see just how high my risk is. Even if I come back showing that, yeah, I could get breast cancer, doesn't mean I'm going to get a voluntary mastectomy. People have done that before, and that's entirely their decision. So we just need to be careful about these things, understanding the system, understanding what may or may not happen, family histories, if you have that family history, and then get those yearly physicals. Anything not normal for you, have looked into. Okay, that's it for this chapter. Hope you enjoyed it, and we're going to move on to the next one.